The discovery of new drugs is a complicated process that takes decades and it costs billions. It's long been the big promise that this process would be simplified with AI. And it already has been, though it's a long road with a lot of variables. Throughout this series, we'll be featuring the stories of breakthroughs and other key moments in emerging tech, as told by those who witnessed them. I'm Jennifer Strong, and this is Shift.
Now here on the steering column is a device called Auto Cruise. You simply set the speed you want.
Self-driving robo-taxis are already on the road in
As the disc rotates, a mirror reflects the light in the way that depends on how the signal was recorded.
This is the 100 terabyte. I present to you Electro, the Moto Man.
Ladies and gentlemen.
I would say that one of my greatest skills is my ability to interact with humans.
This episode, it's the latest installment of our oral history project.
My name is Zhavoronkov, and I'm the founder and CEO of a company called Insilico Medicine. We are a generative clinical stage AI-driven biotech. I am originally trained as a computer scientist, had a brief but very successful career in semiconductors, and then in my early 20s made enough money to think about what I want to do next. And instead of early retirement, I decided to go into biotech and dedicate the rest of my life to understanding why humans age and how to age slower. Started work in generative artificial intelligence around 2015-2016. My first academic paper published in that area was in 2016, showing that we can use generative adversarial networks to generate molecules with the desired properties. That's one of my passions. We demonstrated that we can take those molecules all the way to human clinical trials and go into phase 2 safety and efficacy studies. So we passed safety already. And now we have 31 therapeutic programs out of generative. So I never imagined that I would be able to achieve this just 5 years ago. Originally, we started Insilico in Baltimore at the Emerging Technology Centers of the Johns Hopkins University. So that's my alma mater as well. Some of the original ideas came from my graduate work, and we were trying to utilize deep learning for target identification and disease modeling, trying to understand why disease happens. And my work in aging research helped us a lot at the time. So we started training deep neural networks to predict human age first using many, many different data types, mostly gene expression, protein expression data, methylation, and other data types that are nowadays called omics. So different data types that change in time and are related to your health in one way or another. And we started building age predictors first because everybody in the world has age. So you and I, we both have age in the passport, right? And it actually is a very strong feature which correlates with when we're going to die, because humans have limited lifespan, and that's what we really want to research. And if you want to understand the basic biology of disease, you really want to understand the basic biological concepts of aging. So if you train deep neural networks to predict age on massive data sets, again, everybody has age, you train deep neural networks to understand basic human biology, and then you can retrain those networks on diseases where you have much lower number of examples compared to, everybody. And deep neural networks try to understand which factors are present in age and disease at the same time, and which factors are causal. What is leading to the break in the system that leads to the disease, and then how does this disease progress? So using this approach, we built massive deep neural networks early in 2014, 2015, which was 10 years ago. Can you imagine? And started looking at which features are most important in aging and disease at the same time, and started presenting our work. At that time, as I mentioned, deep learning was a very exotic technology. So every time you publish anything in a peer-reviewed journal, you need to prove that it outperforms classical statistical methods. And you had to fight for every research paper, and we managed to publish a bunch. So we started as a target discovery company, trying to understand the mechanisms of disease and what is driving the disease using deep learning. And then we realized that there is little money in this area. So you could partner with pharma to help them solve diseases. They're not willing to pay a lot of money for that. And also they learn from you very quickly. Then they hire a bunch of people to do the same thing, and you lose revenue streams and you cannot sustain your research. So we decided to go into chemistry. If you own your molecule in drug discovery, and if the molecule is novel, it is selective, it only hits the target that you wanted to hit, and it is safe, and also the target that you chose, think of it as a lock and key paradigm. So you need to find the perfect lock and then find the perfect key that fits that lock. So and the lock needs to be opening the right door, so to speak, that will solve your problem, where you have the prize. So imagine that there are like 10,000 doors with different locks. You need to find the right one where the prize is. Then you need to find the right key for that lock. So in drug discovery, the ownership of the molecule is much more important than the ownership of the target, because you cannot really patent the target. And we decided that we will have the end-to-end system where we identify the targets and make the molecules. And at that time, that was actually a really fun time, 2014, Ian Goodfellow and Yoshua Benjo out of the University of Montreal. Later, Ian went to Google, and after that to OpenAI. They published a wonderful new technology called Generative Adversarial Networks, where two deep neural networks are competing with each other. One is generating deep fakes, so to speak, in response to some generation conditions. So imagine that you say that I want to see a pink bird. So it starts generating a bunch of those birds. And the second network is a discriminator. It's trying to identify whether the output of the first deep neural networks is true or false. So together, they compete with each other. One is trying to trick the other to believe that the content is true, and the other one is trying to get better at identifying fake content. So over billions of iterations, those adversarial networks start generating really believable high-quality content, so high-quality data. And we thought, okay, well, where would this technology work for us? We saw that people started producing really high-quality deepfakes. So you could, generate Brad Pitt, who would be 70 and at the same time have Asian features and in glasses, without glasses, etc., large distributions of that. So we thought that, okay, we're going to first try to use it for generation of high-quality synthetic biological data. So instead of trying to take data from humans, how about we generate really good fake human biological data with the desired conditions? And guess what? Using age as a generation condition. So now, just what I've described, where we use imaging data, like a picture of Brad Pitt as a template, make 1 billion Brad Pitts in the future from 70 to 80 years old, with different input parameters to see how different input parameters change that person in the future. So we can do the same thing with biological data types that are not easily understandable and comprehensible to human eye or mind. So things like gene expression data, protein expression data. So we started creating billions of fake humans in the future and see which features lead to cancer, which features lead to diabetes, etc., and started using that for target discovery. And at the same time, again, we had this need to own our own chemistry. We thought, well, can we actually apply that to chemistry? Can we make deep neural networks, generative adversarial networks, imagine new molecules from scratch with the desired properties, with the properties of perfect drugs, instead of searching for those molecules in libraries that everybody else has access to, and they have been tested many times over. And again, nature did not make every possible molecule, so it's very difficult to find those great molecules that make perfect drugs. A group from Harvard led by Alana Spurogudzik, a genius scientist. At that time, he was a fierce competitor. But then we actually partnered with Alan and tried to do an experiment where we can do a race and showcase to everybody, to all the skeptics in drug discovery, that generative adversarial networks can truly work on a real case study where we get protein target, make our generative tensorial reinforcement learning system imagine new molecules with the desired properties, then synthesize and test. And actually test whether those molecules have the properties. By that time, we already had a few experimental papers, but wanted to make a very clean-cut study and time it. And we partnered with a company called Wuxi AppTech. It's the largest contract research organization on the planet, to my knowledge. The industry, biotechnology industry, changed in a very similar way as IT. So Apple does not make its own computers, it outsources to Hon high precision industry in China, quite a bit of that. Biotech is similar. So if you want to synthesize, toxic chemistry and then test it in cells and animals very, very quickly, you just outsource that. You send your design to the blueprint to the contract researcher, and they will pay, and they will give you the data back. And very, very quickly, they picked an arbitrary target. It was called DDR1 kinase, famous target in cancer and fibrosis. And we gave it to our Gentrol system, Generative Tensorial Reinforcement Learning. In 21 days, it generated 6 molecules that were immediately synthesized by Wuxi AppTech. We tested 6 of them in enzymatic assays, meaning trying to see if they bind to the protein of interest. 4 of them worked. We were like, wow. Then we gave those molecules to a different contract researcher to do metabolic stability and microsomal stability assays, where you actually test whether the molecule degrades really quickly, right? If it degrades very quickly, it's not going to be a good drug. And boom, 2 of them worked extremely well. And then we tested it in mouse. We tested 1 because it actually costs a lot of money to synthesize those molecules from scratch. And it worked in mice, showed really good pharmacokinetics properties, and all that in 46 days. So usually it takes about a year and a half to do that. And yes, we had to cut corners, but 46 days out of generative AI. And we published a paper. We submitted the paper to Nature Biotechnology in 2018 in the fall. Only in 2019 in September it got published. So it took almost a year to publish. For everybody in the audience, it's a very painful, tedious process to convince the expert audience that your technology is worthwhile being published in a peer-reviewed journal. But you still should do that, because otherwise you're just making claims that are not validated by the community. And once this paper got published, everything changed. At that time, a lot of old school computational chemistry scientists that are actually, I admire some of them, they started criticizing that paper, saying that humans also could do that, or maybe they are not exactly super novel, or maybe AI picked very, very similar ideas from humans, and they didn't really think about the algorithm, right? And they didn't really think what we did. They looked at the molecules only. And that's what medicinal chemists do, computational chemists do, right? So they don't think about how it was done. They usually look at the end result, which is correct, right? Because you've got 12 years ahead of you than validating it in humans. But at that time, I was feeling very sad every time somebody published an article saying that, oh, AI is not there yet. Around 2019, we decided to also launch the software to the market. So we said, well, why don't we actually make it available to everybody? And, you go and use it. Doing it yourself, you can use multiple generative engines that we developed, plug in your own if you like, if you think it's better. And then 10 out of the top 20 pharmaceutical companies have used my software in one way or another. And this ability to create a community around the software enabled us to develop a new technology that we call reinforcement learning from expert human feedback. So now when a lot of people use your tools, just like with the case of OpenAI ChatGPT, that's what made them great, reinforcement learning from human feedback. But they paid people $2 an hour to get the feedback, to evaluate the output of generative. We cannot pay $2 an hour to expert chemists or biologists. Those people are the smartest people on the planet. You cannot even hire them on contract. They have to be using the tools for something useful to them. And they usually just like or dislike the output of our platform. And we train the platform on that feedback. We basically either kill some of the generative algorithms if people don't like the output. We cannot see what they do. We are restricting ourselves from getting access to their data, believe it or not, unlike many other AI companies. I have a policy against that. And we only ask them to rank the performance of each model. And now our models get better and better every hour, which is amazing. So nobody else, to my knowledge, has this capability, but that wasn't enough. People are not using our tools to the limit. And in 2019, we decided to raise quite a bit of money from expert biotech investors to kickstart our own drug discovery programs, to actually use our software to design our own molecules and try to go into human clinical trials. And at that time, we also realized that that's where the money is. And if we want to fix the pharmaceutical industry, we should not really try to enable the pharmaceutical companies to do what we do. We do that anyway. They are generally slower for many other reasons, also because of the way they are run. And we decided to develop our own pipeline of therapeutic programs. So for that, I also realized that I wouldn't be able to do it myself. I want to achieve the level of quality in our molecules that goes above and beyond the level of quality in the pharmaceutical companies. I want to be able to impress the Japanese biotech. And I thought, okay, well, how do I do that? So first, I decided that we need to ensure that if we are subcontracting contract research organizations, we want to be there. And since 90% of them are in China, we actually expanded in China as well to have our own people supervising the scientists at those CROs, contract research organizations. So they manage the IP properly. We also get the best of the best people, get the best chemistry cooks. And we also ensure that the data that they generate for our own programs goes back into AI and nowhere else. While working with some of those CROs, I met a wonderful scientist, somebody I deeply admire. His name is Dr. Ren Feng. I basically started working with him on a daily basis while he was cooking my chemistry and helping me with biological experiments. And at that time, you remember COVID hit, so 2000, right? In China became closed, right? So I had to travel there from time to time, and I spent a lot of time in quarantines. I spent like 18 weeks in quarantines. That's how I actually built a little bit of muscle. So I just didn't have anything else to do, just, do push-ups. I had a chin-up bar, so chin-ups, and a lot of conference calls. And Dr. Ren, at that time, became our chief science officer. So he joined us in 2021, in January, and that's when the company changed. So we realized that a person who is super expert in drug discovery and can run a large science organization should be responsible for drug discovery. So we decided to have me in charge of AI and Dr. Ren in charge of drug discovery. And we started setting, to my knowledge, again, I have to make all those disclaimers, we started setting industry records, not only beating records in science papers, right, 46 days to mouse, we started going to humans, right, all the way, and trying to generate really ultra high quality drugs from scratch for moderately or highly novel targets. So one of the problems in the pharmaceutical industry is that everybody is going after the same thing. So with AI, we could actually get to a little bit higher level of novelty, but not too high, because too high means that nobody's going to buy your molecule until you complete the phase 2 clinical study. It's very risky. So we found an optimal balance and started moving. And in 2021, when Dr. Rand just joined us, we nominated 2 preclinical candidates. So here I think I want to deviate a little bit and explain how drugs are discovered. So usually in the preclinical world, before you go into humans, that period takes you about 6-7 years, sometimes much longer. It starts with understanding the disease hypothesis, understanding which protein target is driving disease, narrowing down the disease to just one target or a couple or maybe 3 or 4, and then validating that this target works. So you actually need to perform a lot of biological experiments to convince yourself that you want to bet on this target. But usually this is a molecular casino. You want to bet a lot of money on one target, and you don't have a lot of bets to make, right? Usually you're very restricted by financing. Then you go and identify small molecule hits or antibodies that disable that protein. And that process takes about maybe a year and a half. Usually it's successful, but then you start improving these molecules, usually about a year and a half to 2 years, and that fails a lot. And then you do additional preclinical studies to nominate the preclinical candidates. So you do animal efficacy experiments. Then you do toxicity studies, and then you go into human phase 1. You test safety in humans and healthy volunteers usually, or in cancer in patients right away. Then phase 2, it's safety and efficacy, and phase 3, it is a very large scale study. That also takes you about 6 to 7 years. So it takes you about 10 to 14 years to discover and develop a drug. But the first stage is very important. If you get it wrong, you fail later in phase 2 human clinical trials, and you lose hundreds of millions of dollars and also a lot of human work. So you need to be damn good at these first stages. So we started nominating those high-quality preclinical candidates so one notch before human clinical trials. And in 2021, we nominated 2. 2022, we nominated 9. 8 internal, 1 for a partner. Just to put you in perspective, big pharmaceutical company, and I'm not going to throw names, but let's say top 10. Within top 10, you would nominate 4 to 5, usually, after spending hundreds of millions, if not billions, on R&D at the preclinical stage or research. So nominating 9, that started getting people's eyebrows like, wow, okay. And many of those targets are novel or moderately novel. So that makes it even more difficult to do the chemistry because there's not much known about the target. Then we started going human clinical trials. So 2021, we went first in human. 2022-23 completed several clinical trials and safety in New Zealand and in China. And 2023, we entered phase 2 human clinical trials. Also separated in the US and in China 2 parallel clinical trials. Just because, nowadays I'm very unhappy about this because all of us are on the same boat when it comes to health, but geopolitical arguments that politicians make introduce some new risks, right? And we want to be prepared for an all-out, economic or whatever war. So we try to ensure that we are ready for all kinds of scenarios, even though I think that right now we really need to advocate, all of us need to pause and think about how to maintain peace. Imagine that we're in cookie business. So you can either sell the software to make better cookies and to forecast which cookies are going to be more popular in the future. You can collaborate with cookie sellers, cookie makers, integrated systems, big pharma in this case, to help them bake the cookies and take a percentage. Or you can bake your own cookies, and then the cookie sellers will actually buy it from you if the product is very good because you found the flavor that everybody likes. They would want to have it and they are willing to pay top dollars for it. So we started doing all 3. But we value the last model where we actually are the cook more because that gives you much bigger impact on the industry. We want to become the cornucopia of ultra high quality drugs for the pharmaceutical industry. And they're facing a shortage of good drugs. Currently, we are going to be flooded by the wave of expensive drugs. We are already under the water, and we need to make those drugs cheaper, want to make them more accessible, and also go even after rare diseases. Rare disease patients are not serviced at this point in time. And when it comes to aging, you and I were both patients. We're working relentlessly towards this goal. I dedicate my entire life to this. I don't have a family, I don't have kids, I don't have many friends. So I sleep 4 hours a day, and that's my only goal in life. And many people who work with me work my hours.
This episode was produced by me and Emma Silikans and mixed by Garrett Lang, with original music from him and Jacob Gorski. Thanks for listening. I'm Jennifer Strong.
From PRX.